We've seen how DNA is packaged in eukaryotic cells, but we've spoken about DNA as if it were a constant entity. But in fact, DNA sequences change over time, and they can change very significantly. The DNA in each of our cells is probably different because these sorts of changes have occurred. These alterations are not designed, but they occur in single base pair changes or in small segments because of mistakes in the replication, the copying of DNA, or because of damage to the DNA that escapes our many repair processes. More on that later. There are changes that occur accidentally, for example, exposure to UV light or radiation, or in response to environmental conditions. An example of the latter is certain types of contaminants in the environment that are carcinogenic or mutagenic that impact DNA base, structure, or the overall structure of DNA, the phosphodiester bond. Large-scale movements of DNA also occur, sometimes by errors or elicited by specific entities that are incorporated into the DNA called transposable elements that move on their own and take genes with them, and we'll talk more later about these elements. Transposons have overall had a significant effect on DNA sequence. The processes of duplication and divergence are very important because these mechanisms provide a way of generating new functions. Gene duplication, for example, occurs pretty much randomly, and therefore the DNA undergoes changes in either or both of the genomic elements that result that in turn give divergence in function. There is no pressure on a duplicate gene to perform the same function, so its sequence may change and still retain the same function, or may change and become non-functional, called a pseudogene. Or the change in sequence can generate a new function. In this case, the original function is maintained by the original intact gene. There are multiple examples: receptors, regulatory proteins, globins. But globins are a great example, and we'll focus on hemoglobin that in our blood. An early globin gene that bound oxygen underwent duplication, and the alpha chain gene and the beta chain gene of hemoglobin emerged, similar in function, binding oxygen, but different in sequence. These genes that diverged are called paralogs. Over time, these monomeric proteins acquired the ability to assemble into tetrameric hemoglobin. Genes that produce the same function in different species are termed orthologs. And all of these different but related globin genes are homologs. Note that orthologs are the alpha chain in different species. Paralogs are the alpha chain and the beta chain, and the entire family is homologs. So how can divergent functions be generated? One mechanism to create multiple functions from a single coding sequence is to recombine coding regions. We start with 4 different coding regions, and generally genes that are present and have survived in an organism will be required, so a change that renders them inactive will be lethal. However, if there are gene or chromosomal duplications, now there are 2 copies of each region, providing the potential for divergence while keeping essential functions. There are multiple possibilities for next steps: combination of 2 coding regions in a way that confers new functions that may be beneficial to the cell. Completely new functions can be derived from the prior sequences, or the duplicated DNA itself can be lost and become non-coding and non-functional. Changes in DNA are part of generating both new functions and new species. A single species may diverge into 2 different species because of the kinds of changes that we have discussed by small duplications, mutation, or recombination. But remember that changes in DNA can also be fatal, so extinction can occur if mutation happens in an essential gene. Interestingly, genome duplication events are common in plants and fungi, where the entire genome is duplicated, and even the vertebrate zebrafish appears to have undergone this type of event. During replication, chromosomes do not separate into two separate cells but stay in the same cell, so the entire genome is duplicated. In some cases, that can happen more than once, yielding higher order numbers of chromosomes and allowing still more differentiation. Following duplication, divergence of function and even new functions can arise with recombining different genes and even yield a new species. So the genetic material of living organisms is not as permanent as we might think, with additions that arise from duplications and from transposon action. Deletions occur spontaneously in response to DNA damage and other factors, but some conservation must occur. For example, mammals have a very similar genome size, whereas other organisms have quite wide ranges. But of mammalian genomes, only 150 million base pairs comprise the coding sequences, a very small fraction of our total DNA sequence, and many changes occur in non-coding DNA. Most vertebrate genomes are smaller than mammals, but the largest genome known is for locusts. So there is substantial variation, and care must be applied to making predictions regarding genome size. The variation in insects from 130 million base pairs to 650,000 million base pairs illustrates how significantly genome size can vary. DNA sequences themselves are highly informative, and we'll learn about DNA sequencing methods as part of homework in week 4. Some variations, even within a single species, involve large regions of DNA, whereas others may be only a single base pair change that encodes a totally inactive protein due to a change in an amino acid crucial to function of that protein. These single base pair changes are termed single nucleotide polymorphisms, or SNPs, because that position varies across a population or between species. Using these SNPs and larger sequence changes, it has been possible to link DNA sequence to disease in a growing number of cases, with new additions to the list almost daily. Our ability to determine DNA sequence allows us to compare this genetic information across organisms as well as between individuals. It is interesting to compare human and ape sequences, as what we see is drift in the sequences, with strong similarity and some random differences, and perhaps some very specific differences, as we'll see. The DNA sequence of humans and apes is approximately 97% identical. Even the genes expressed are similar in liver and in blood cells. So where are the differences? The significant sites for variation between apes and humans are in regions involved in neural functions. And as sequences vary more in these areas, we can see at least 5 times different amounts of proteins being produced in brain cells versus the much smaller differences observed in liver and blood cells. So do you think that the differences in sequences, and perhaps importantly in the relative gene expression in the brain, suggest an essential role in regulation of protein expression and outcome for apes versus humans?